it's great to follow on from this from the great talk by Rafael. I'm going to talk a little bit about similar things. In particular, an autocatalytic reaction network that produces nucleotide precursors. Okay, so I'm a prebiotic chemist, and the type of questions that I think we're all interested in is which is about Earth 4 billion years ago or so, may as well have been an alien planet compared to today, to going to a planet like this, where everywhere we look, we see life, more or less, especially water elements. Question, of course, and when it comes to a definition of life, we can't even really agree on that. That's how difficult the problem is. But the definition that often comes up is this one, NASA's working definition: life is a self-sustaining chemical system capable of Darwinian evolution. So Darwinian evolution implies that you need to have molecules or systems of molecules that undergo replication. That's the big question: how do you get molecules to undergo replication so you can have Darwinian evolution? Well, one solution to that could be this idea of autocatalysis. And that's where this model for the chemoton comes in. So the chemoton is short for chemical automaton, or like little chemical robot. And it was developed by Tibor Ganti back in the 70s. And so without specifying any particular chemistry, what this model says is all you need is, in principle, 3 autocatalytic cycles. You make a simple cell, protocell, if you will, that would be capable of undergoing Darwinian evolution. It could reproduce itself in an adaptive environment. So, but you need at least these 3 metabolic cycles. So the one of them is the genetic polymer cycle. So that's things that we think of like DNA in particular, represented by this little loop here. The membrane cycle. So this is the cell is composed of some sort of membrane. Oftentimes it's either thought of as fatty acids or phospholipids of some sort. We need some sort of autocatalytic cycle that produces the membrane. And then you have this metabolic cycle, which is basically feeding production of intermediates for both the template cycle and the membrane cycle. And so what I want to focus on today is just this kind of metabolic cycle here. Is there any way we can try to come up with something that would be consistent with this model that we could learn from? Okay, so what is autocatalysis in the first place? Strictly speaking, the definition is isothermal acceleration of the rate of a chemical reaction, the products of this reaction. It has a pretty, mathematical definition shown here. The rate is proportional to this autocatalysis there. And so this leads to this kind of signature, this auto amplification signature, where you start with a little amount of autocatalyst. It eventually starts to increase, and then you enter this exponential phase. Then as your starting material runs out, you go to the saturation phase. So this is kind of what people typically look for when they're looking for an autocatalytic reaction. And so why is it special? Well, here's another way to look at autocatalysis. It's a way to have a chemical reaction sort of copy itself. So here's one copy of A, takes n number of C, converts it to A. So you start with 1, end up with 2. This is the process of DNA replication is autocatalytic in principle. And if you really zoom out and look at the whole replication process of the cell, it can be thought of as just one complex autocatalytic reaction going on to give you that step. So intrinsic to biology. A lot of people think it's really important in the emergence of life on Earth. So, one possibility for autocatalysis on the Earth that's been studied quite a bit is the formose reaction. That's just shown here, kind of in simplified form. So what is the formose reaction? You just start with formaldehyde, a little bit of glycolaldehyde as your autocatalyst. You need high pH and some calcium as a catalyst. And then you can make 2 copies of glycolaldehyde, for example, but you also make a whole bunch of other sugars. People have, including ribose, which people have claimed would have been useful prebiotically. So you can get formaldehyde from a variety of sources. There's this report in Science. You can get it from CO2 and water, radiation in the atmosphere. You can get it from Miller-Urey type experiments, and even impact syntheses, for example. So formaldehyde's pretty reasonable starting material, if you will, on early Earth. And so here's kind of what's known as the Breslow cycle. A lot of people, disagree whether this cycle is really exists or not, but it's just kind of an easy way to explain the formose reaction itself. So you just start with glycoaldehyde. You do an aldol addition with formaldehyde to give you glyceraldehyde. You isomerize to dihydroxyacetone, do another aldol addition, another isomerization to this tetrose, and then from there you can get fragmentation into these two copies of glycoaldehyde. And of course, you're doing a lot of other side reactions on outside of this cycle, which are giving you these higher order sugars. So that's just a simplified version. Okay, so we thought, well, maybe is it possible that this formose reaction can kind of serve as this metabolic cycle in the context of this chemoton model? So that means it would have to it would have to feed into this template reaction, for example. And that's the first thing that we wanted to test if that could be possible. So is it possible to couple this autocatalytic reaction with production of genetic polymers? And of course, now we're just relying kind of on RNA world hypothesis. I suspect most people know what that is. But it basically, RNA is the strong version of the RNA world hypothesis, suggests that RNA was actually the original genetic polymer to arise chemically on early Earth, and that gave rise to the cells, all the cells that we see today based on RNA. That's what we were going for, and so we wanted to see if we can couple this cycle to the production of ribonucleotides in particular. So we needed to rely on some reported prebiotic syntheses. And of course, Orgel showed back in 1970, simplest thing you could try is just take some D-ribose, try to condense it with pyrimidine, cytosine, and to make a nucleoside, but this reaction doesn't work. So then in response to that, he came up with this other pathway, Orgel and coworkers, where they take D-ribinose with cyanamide. And this is, some of this work what I'm about to show was actually talked about by Rafal previously, which is great. So he took this cyanamide to react with D-ribinose and made this aminoxazoline intermediate, and then cyclized with cyanocetylene, followed by dry down to get this 2,3 cyclic 2'3' cyclic cytidine phosphate. And then Sutherland and Pauner came along, and this pathway is commonly known as Pauner-Sutherland pathway. And I think it's great. I mean, it just shows like how influential this research has been, especially this paper, I would say, that it just impacts so many people's research. But this pathway is even simpler than the one that Orgel showed, because you can start with this C2 instead of a C5 sugar, if you will, react with cyanamide to make this intermediate, and then react again with glyceraldehyde to give you the same aminoxazoline. So what we wanted to know is, can we integrate one or both parts of this pathway with the formose reaction? It's a simple question that we wanted to see if it was possible. So this is work done by my student Jeremy Tran at UNSW. So all we did was, we took the formose reaction and just added cyanamide to it, with the idea that, well, we would make 2-aminoxazole and potentially even the 2-aminoxazolene stereoisomers through a couple of these different pathways. One is kind of the Pauner-Sutherland pathway, the one is the Orgel pathway. And maybe if we're super lucky, we can get all the way to RNA, but there's some dotted arrows there. That's kind of just the idea of the proposal. So first we looked at the NMR just to see if we could actually make 2-aminoxazole. So all we did here was just take some formaldehyde, a little bit of glycoaldehyde, reacted under formose conditions with cyanamide initially added. And you can see in the NMR at the beginning, we see just some acetate acetate from the calcium, methanol and formate from K0 reaction. But then after 22 minutes, you can see these peaks for 2-aminoxazole, and we measure a 17% yield, which is not too bad, for just kind of a prebiotic reaction, I would say. Okay, so we can make it, but we're also interested in the kinetics of the formose reaction and what's going on from that perspective. So like we were saying before, when you have this autocatalysis, you kind of have these three phases. So the first phase is this lag phase, so you have lots of formaldehyde. And then as the formaldehyde gets consumed and converted into autocatalysis, you start to enter this exponential phase where it very rapidly decreases. And then you eventually enter what's called the yellowing point, and then this so-called degradation phase after you run out of formaldehyde. And you start making what's known as a tar-like mixture. It turns yellow, kind of brown, caramelization. Okay, so we wanted to know if the nucleotide precursors, the 2-aminoxazole that we're seeing, kind of follows the same trajectory or not. So here's the experiment that we did. Just added different amounts of cyanamide to this formose reaction, and then we quenched it and derivatized with this DNPH chromophore, so we could analyze by HPLC. So with 0 millimole cyanamide, you can see that the formaldehyde pretty quickly is depleted, and then you enter into this kind of degradation phase really fast. This graph here just shows kind of the same thing, except it's looking at glycolaldehyde concentration. You get a nice peak, and then as we enter into the yellowing phase, it starts to go down. When we add more cyanamide, we can actually see that the lag phase starts to increase. So the increasing cyanamide concentration increases the lag phase, and you can see that reflected here in the delay of the glycolaldehyde peak as well. And if we go to 9, we increase that lag phase even more. Okay, so what's interesting here though is although cyanamide increases the lag phase, it doesn't completely shut down the autocatalytic reaction. It just modifies the kinetics. And if you add even more than 9 millimolar, you can get really long extension times, but we couldn't really reproduce them very well. So what we think going on, is just you have cyanamide there. It's stealing away some of the glycolaldehyde, as well as these other sugars as part of this minimal cycle. It's just consuming some of these autocatalysts and converting them to this 2-aminoxazole, and as well as other derivatives. Okay, so we wanted to see if autocatalysis was actually the main pathway, because you might think, well, if you're starting off with glycolaldehyde and cyanamide, you should no doubt expect 2-aminoxazole. What we wanted to know is most of that 2-aminoxazole actually coming from glycolaldehyde produced from the formose reaction, and not the glycolaldehyde we initially added. We used this isotopically labeled experiment to find out. So this is the non-labeled 2-aminoxazole that you would expect to see just from this initial glycolaldehyde, and you see a little bit, but you see most of the doubly labeled that you get from the formose reaction as the product. So this shows that the 2-aminoxazole is predominantly formed from the formose generated glycolaldehyde, and that again the yield is about 17%. So just as this cycle converts more of the sugars into these 13 sugars, we can get that 2-aminoxazole. So we had to use LCMS, look at 2-aminoxazoline production, this one right here, and this time we use 13C glycolaldehyde, and we see a similar trend. So the initial 2-aminoxazole that we add, the aminoxazoline that we see produced is mostly formed from the formose reaction, the glycolaldehyde produced from. Okay, so here's kind of a more complete picture. I'm kind of running out of time. So this is just by no means the full story. This is a very simplified scheme of what we think is going on. But as it turns out, we are making these pentose aminoxazolines We think most of it is actually going through this kind of organelle pathway as opposed to this amino-oxazole pathway that combines with glycerol. Most of it is through this direct reaction with pentoses with cyanamide. Okay, so in conclusion, the autocatalytic formose reaction can fuel the prebiotic synthesis of these nucleotide precursors in a manner that is reasonably consistent with chemoton. And the synthesis of these nucleotide precursors is kinetically coupled with the kinetics of the formose reaction. So this is really interesting for us because it has the potential for some interesting feedback capability. And in other words, cyanamide peels off excess glycolaldehyde, but it doesn't actually disrupt the cycle. The cycle just keeps on going. It just kind of slows it down initially, but it doesn't completely stop it. And there are still a number of challenges, of course. Selectivity. There's so many other products for being made. They're going to be a problem. The high pH we need is not really compatible with nucleobase pairing, or even hydrolytic stability for that matter. And under the pH conditions, the 2-aminoxazole is not directly used. And will the other steps in this counter-cellular pathway actually work under conditions? So, still a ton of challenges, by no means done. But this is kind of the stuff that we're interested in right now. I just want to acknowledge Jeremy Tran in particular. He did pretty much all the work that I just showed, as well as other members of my group in some of these funding sources. Thank you.